Hello friends, welcome to my channel. Today is my first video and my name is Ari Prabhagaonkar, studying in standard 12th, and we'll be doing the biotechnology chapter 1, that is the biotechnology, its principle and processes chapter. We'll be reading NCERT line by line and I'll be telling you few important things which you have to mark on your NCERT. So please keep your NCERT book ready with you, or if you have PDF then keep it ready. And as you are new to my channel, please subscribe my channel and click on the bell icon. So let's begin. Biotechnology. In Unit 9, there are two chapters of biotechnology in Standard 12. Out of that, we'll be doing the first chapter, that is biotechnology, principles, and processes. So, what is biotechnology? Biotechnology, it deals with techniques of using the live organisms or the enzymes from organisms to produce products and processes useful to human beings. So, I'll mark this statement here. Just a minute. Yes. So biotechnology it deals with the techniques of using live organisms or enzymes from microbes from organisms to produce products and processes useful to human beings. So please mark this on your NCERT. Okay, and in this sense, making curd, like even bread or wine, you must have heard about the yeast used in fermentation while making the bread or wine, which are all the microbe related or mediated processes. It could also be thought as a form of biotechnology. So, however, it is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on the large scale. Okay, so now this definition is uh, it's not complete and it's not sufficient. So that's why the European Federation of Biotechnology has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. And the definition says the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and processes. So the definition is here: the integration of natural science and organisms, cells, parts thereof, and molecular analogs and products for processes and service. Okay, so now we'll go to the next part, that is the principles of biotechnology. Among many, the two main or the core techniques that enable the birth of biotechnology, the modern biotechnology, are two. That is, one is genetic engineering and the other one is bioprocess engineering. The genetic engineering means the techniques to alter the chemistry of genetic material, that is DNA, in the organisms, or sometimes RNA like in viruses and all, to introduce these into the host organisms and change the phenotype of the host. मतलब जो हम जीन और कुछ निकालते हैं ऑर्गेनिज्म से दूसरे जीन में डालने के लिए, वो हम अल्टर और मॉडिफाई करते हैं. And this process of changing the chemistry of genetic material is called as genetic engineering. Second process is bioprocess engineering. That means maintenance of sterile, contamination-free ambience in chemical engineering process to enable the growth of only desired microbe or eukaryotic cell in large quantities for manufacturing of biotechnological products like antibiotic, vaccine, enzymes, etc. आपने देखा होगा कि आप जब खाना बनाते हैं किचन में, तब we keep the kitchen very clean, and sometimes some people they wear gloves and they cover their head so that the hair doesn't fall. So it comes under the bioprocess engineering, that is maintenance of sterile, contamination free zone. This is very important. Okay, so let us now understand the conceptual development of the principle of genetic engineering. So I'll directly go to this here. The techniques of genetic engineering. The techniques of genetic engineering, which include the creation of recombinant DNA, use of gene cloning, and the gene transfer to overcome this limitation and allow us to isolate and introduce only one or a set of desired gene without introducing undesirable genes into the target organism. मतलब creation of recombinant, पहले recombinant create, after that gene cloning, and then gene transfer. So do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely the piece of DNA would not be able to multiply itself in the progeny cells of the organisms. But when it gets integrated into the genome of the recipient, that is the genetic material, it may multiply and be inherited along with the host cell, host organism, host DNA. This is because the alien piece of DNA has become a part of chromosome, which has the ability to replicate. In the chromosome, there is a specific DNA sequence called origin of replication. Mark this, this is very important. In a chromosome, there is a specific DNA sequence called origin of replication, which is responsible for initiating the replication. Therefore, for multiplication of any alien piece of DNA in organism, it needs to be a part of chromosome, which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host. This is also called as the cloning. So this can be called as cloning or making multiple identical copies of any template DNA. So if you take a DNA or a piece of gene and introduce it into the genome, then it may multiply, and this is called the cloning or the making multiple identical copies. Let us now focus on the first instance of construction of an artificial DNA molecule. So this is not that important, but remember just this thing, that the first recombinant DNA construction DNA emerged from the possibility of linking a gene encoding antibiotic resistance. So I'll mark this. The construction of first recombinant DNA emerged from possibility of linking a gene encoding antibiotic resistance with the native plasmid, that is small circular autonomously replicating extra chromosomal DNA of Salmonella typhimurium. This was the first bacteria from which plasmid was isolated by Stanley Cohen and Herbert Boyer in year 1972, and it was responsible for the antibiotic resistance gene by cutting out a piece of DNA from plasmid, which was responsible for conferring antibiotic resistance. And when you clone any gene, it should be first removed or cut out from the plasmid or its original DNA. And this cutting of DNA was possible because of the specific location. It became possible because of discovery of so-called as the restriction enzymes, molecular scissors. They are also called as molecular scissors or restriction enzymes. They cut piece of DNA, and this cut piece of DNA was linked with the plasmid DNA, and plasmid DNA then acts as a vector to transfer the piece of DNA attached to it. So now we'll go to this part. This ability to multiply the copy of antibiotic resistant gene in E. coli, that is Escherichia coli bacteria, was called as cloning of antibiotic resistant gene in that bacteria. That is, when this plasmid having that antibiotic resistant gene was introduced into the E. coli, it showed resistance to the antibiotic. Actually, E. coli doesn't have any such antibiotic resistance gene in it. You can hence infer that there are three basic steps in genetically modifying organism. The first step is identification of DNA and desirable genes, introduction of identified DNA into the host, and maintenance of introduced DNA into the host and transfer of DNA to its progeny. These three steps are very important, that is identification of DNA with desirable genes, introduction of identified DNA into the host, and maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Now let us talk about the tools of recombinant DNA technology. Now we know from the foregoing discussion that genetic engineering or the recombinant DNA technology can be accomplished only if we have the key tools. That is, the first basic key tool is the restriction enzyme, or you can call them as the molecular scissor for cutting the DNA. Then we require polymerase enzyme for DNA replication. Then we require ligase. Ligases, they do the work of joining the DNA strands. Whenever DNA is cut, we need to join them again. And there we require the ligase enzyme. So the tools which are required are the restriction enzymes, polymerase enzyme, ligase, and vectors like plasmid, and the host organism in which you have to introduce that gene or that particular uh, DNA. Okay, so let's first talk about the restriction enzyme. In 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl group to the DNA, while other cut DNA, and the other one which cut DNA was called as the restriction endonuclease. The first restriction endonuclease was the Hind2. Please mark this. This may come for the NEET or even for boards. The first restriction endonuclease was the Hind2, whose functioning depend on the specific DNA nucleotide sequence, which was isolated and characterized 5 years later. Now you may why it is given the Hind2 and not Hind1, although it was the first restriction endonuclease. It was because this was first restriction endonuclease and not first nucleases. Nucleases have two different groups. One is endonuclease, one is exonuclease. So the first discovered or first isolated nucleases might be the exonuclease, and the second one was the endonuclease. But from the endonuclease category, this was the first in two. And the specific base sequence at which this restriction endonuclease makes the cut or cut the DNA is called a recognition sequence. Besides Hind2 restriction enzymes, today we know more than 900 restriction endonucleases or restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize a different recognition sequence, and it should be a different recognition sequence. They can't have the same recognition sequence. The convention for naming this enzyme is that the first letter of the name comes from the genus. The first letter, uh, the first letter of the name comes from genus. Second two letters from the species of the cell from which they were isolated, and then comes the name of the strain. For example, in ECOR1, E represents genus, CO from species, and R the name of the strain, and 1 represents the order in which they were isolated from the strain. Restriction enzymes belong to larger class of enzymes called nucleases. This is very important. The restriction enzymes belong to a larger class of enzymes called nucleases. These are of two kinds, exonuclease and endonuclease. Exonuclease cut nucleotides from the end of the DNA, whereas endonuclease makes cut at specific locations or specific position. जो एक्सोन्यूक्लियेसिस हैं वो न्यूक्लियोटाइड्स एंड ऑफ द डीएनए से कट करते हैं, एंड एंडोन्यूक्लियेसिस makes cut at specific position. Each restriction endonuclease functions by inspecting the length of the DNA sequence. Once it finds its specific recognition sequence, that is the palindromic sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbone. अगर मान लो ये DNA है, then it will make cut at here like this on both the strands. So each restriction endonuclease, it recognizes a specific sequence. This sequence is called palindromic nucleotide sequence in the DNA. It doesn't cut DNA at any other place rather than the palindrome nucleotide sequence. So here they have shown diagrammatically how it cuts the DNA. Here the vector DNA is given, and here the foreign DNA, and the restriction enzyme with which you cut the vector DNA, the same restriction enzyme should be used to cut the foreign DNA, so that they generate the complementary base pairs or the complementary sequences. Then only you can join the DNA fragments. So when they cut this, they give the sticky ends having uh, only half the nitrogenous bases, and the other half, which are complementary, are present on the other strand, other DNA. So these are then joined, and for joining we use the ligase enzyme. So what is a palindrome? Palindrome is a word which reads same when read from both the parts, from both the ends. For example, Malayalam. When you reverse the order of reading, it reads same on both the sides, from left to right as well as from right to left. And against a word palindrome where the same word reads in both directions, the palindrome in DNA is a sequence of base pairs that read same on the two strands when orientation of them is kept the same. For example, the GAA-TTC, and if you reverse the reading order, it becomes GAA-TTC. This is a palindrome. Restriction enzymes cut the strands of DNA a little away from the center of this palindrome site. Not exactly. So, let's take example of this GAA-TTC. It will not cut the this palindrome at the center. It will make a cut here and here, and it will leave this center part, so that let's suppose if this is the sequence, then it will make a cut on this strand. It will make here and on the other strand it will make it here. In this way it will generate two different strands. One will be like this and the other will be like this. So restriction endonucleases, they are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources of the genome. When cut by the same restriction enzymes or restriction endonuclease, the resultant DNA fragments have the same kinds of sticky ends, and this can be joined together using the ligases. Mark this statement because this is very important and this might come for NEET. जो आप रिस्ट्रिक्शन एंजाइम यूज़ करते हैं वेक्टर को कट करने के लिए, उसी रिस्ट्रिक्शन एंजाइम से आपको फॉरेन डीएनए कट करना है, तभी सिमिलर टाइप ऑफ स्टिकी एंड्स जनरेट होंगी. इफ दे आर नॉट सिमिलर, देन you will not be able to join them using the DNA ligase. They should be similar or complementary to each other, and then only you can join them end to end using the DNA ligase. So they have shown here diagrammatically. Here the foreign DNA is cut and the vector is also cut by the same restriction enzyme. So they have the complementary sticky ends which are then joined by ligases, and this recombinant plasmid or DNA is formed. The process of introduction of this recombinant DNA molecule into the bacteria is called as transformation. So this process is called as transformation. And those bacteria having this recombinant DNA molecule in them, which we had inserted, are called transformants. So separation. Now let us come to the separation and isolation of DNA fragments. The cutting of DNA by the restriction endonuclease results in the fragments of DNA. So these fragments can be separated by a technique known as the gel electrophoresis. So in a mixture, there will be a lot of different fragments of DNA cut by the restriction endonuclease, and we have to, before joining them with the ligase, we have to separate this by a technique known as the gel electrophoresis. Since the DNA fragments are negatively charged, मतलब जो DNA fragments होती हैं उन पे negative charge होता है, and they can be separated by forcing them to move towards the anode under the applied electric field through a medium or the matrix. In gel electrophoresis, there are two electrodes. One is anode and cathode. Anode is positive, cathode is negative. So, since DNA molecules are negatively charged, they are forced to move towards the positively charged anode. They move towards anode under the application of this electric field, and when they move towards anode, they don't move simply. They require a medium or a matrix to move. And nowadays the most commonly used matrix is agarose gel. Mark this because they have asked this for many competitive exams, and this might come for the NEET as well. So the most commonly used matrix for gel electrophoresis is the agarose gel, which is a natural polymer extracted from the seaweeds. It is like the agar-agar, which is obtained from the red alga, just like carrageen and the algine from the brown algae. We get agarose, which is a natural polymer extracted from the seaweeds, and these DNA fragments separate according to their sizes through the sieving effect provided by the agarose gel. मतलब जो साइज से बड़ी होंगी they will take time to cross the matrix, so their movement will be very slow, and those which are very small they will move faster. So smaller the fragment, farther it moves. Smaller the fragment, the farther it moves. Mark this. This is very important. The separated here, see in this diagram they have shown the DNA gel electrophoresis method. The larger fragments are behind and the smaller ones are further. So smaller the fragments, farther it moves. And the medium is this, the grayish material which they have shown is the medium, agarose gel. In the diagram they have shown it as a grayish color, but in reality I don't know what is the color. It may be a red color. The separated DNA fragments can be visualized only after staining the DNA with a compound known as the ethidium bromide. Mark this. This was there in NEET. Ethidium bromide is a compound which is used to stain these DNA fragments. You can't see them with your naked eyes and in visible light. They have to be stained with the ethidium bromide and then followed by exposure to UV radiations. So you can see the bright orange color pens after staining with ethidium bromide followed by exposure to the UV radiation. अगर मान लो आप एक माउंटिंग कर रहे हैं सेल का, तो उस माउंटिंग को आप you can't see them with your naked eyes. You have to stain them first with methylene blue or acetocarmine stain, and then you have to see them under microscope. Similarly here you have to stain them with ethidium bromide followed by exposure to the UV radiation. उसके बाद आपको वो ब्राइट ऑरेंज कलर्ड बैंड्स ऑफ DNA दिखेगी. So you will see a bright orange colored bands of DNA in the ethidium bromide stained gel exposed to UV light, and now the separated bands of DNA are cut out from the agarose gel. मतलब जो जेल पीस में जो DNA fragments हैं they are cut out from the agarose gel and extracted from the gel piece. This step is known as the elution or gel elution, and the DNA fragments are purified in this way. They are used in the construction of recombinant DNA by joining them with cloning vectors. So first what we did, we used the restriction enzymes and we cut the foreign DNA with this restriction endonuclease, and it made a lot of fragments. Then we separated the fragments using the gel electrophoresis method. After that we stained them with ethidium bromide followed by exposure to UV radiations. Then we could see them as the orange colored bands. After that they were removed from the gel by gel elution process, or simply you can say elution. And now we can use them for the recombinant DNA technology, like for joining them to the plasmid. Cloning vectors. You know that plasmids and bacteriophages have the ability to replicate within the bacterial cells independent of the control of chromosomal DNA. Bacteriophages are the viruses those are the viruses that infect the bacteria. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cells. Some plasmids, they may have only 1 or 2 copies per cell, whereas others may have 15 to 100 copies per cell. It depends on the organism and the cells. So their numbers can even go higher. If we are able to link an alien piece of DNA with bacteriophage or a plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. So vectors used at present are engineered in such a way that they help us in easy linkage of the foreign DNA and selection of recombinant from the non-recombinant. So जो एंटीबायोटिक रेजिस्टेंट जीन हम इसमें डालते हैं, they help us in selection of recombinants from the non-recombinants. This will be done in the next section. So the following are the features that are required to facilitate cloning into a vector. The first is origin of replication. The plasmid or the vector which we use it needs to have a origin of replication. This is a sequence from where the replication starts, and any piece of DNA when linked to this sequence, it can be made to replicate within the host cell. This sequence is also responsible for controlling the copy numbers of the linked DNA. Okay, so this is very important. The sequence which initiates the process of replication is the origin of replication, from where the replication starts, and any piece of DNA which when linked to this sequence can be made to replicate within the host cell, and this is also responsible for controlling the copy numbers of the linked DNA. So now we'll go to the next part, selectable markers. So I told you about antibiotic resistance chain, which are actually the selectable markers. In addition to ORI, that is origin of replication site, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. So what are transformants? See, transformation is the process through which you introduce the DNA into the bacteria, and those bacteria having those introduced DNA in them are called as transformants or transformants, and those bacteria which lack the introduced DNA or which do not take the introduced DNA in them are called as non-transformants. Transformation is a procedure through which the piece of DNA is introduced into a host bacterium. Normally, the genes encoding resistant to antibiotics such as ampicillin, tetracycline, chloramphenicol, or kanamycin, etc., are considered as useful selectable markers for E. coli. The normal E. coli do not carry antibiotic resistance. Just mark this, this is very important. Normal E. coli cells do not carry resistant against any of these antibiotics. अगर आप नॉर्मल E. coli लें और उसको एंटीबायोटिक्स में डाल दें तो वो मर जाएगा, क्योंकि इट डजेंट हैव द नेचुरल resistance against this antibiotics. But if you introduce them antibiotic resistant gene in them with the help of vector, then they can show the resistance towards the that specific antibiotic which you have introduced. Cloning sites. In order to link the alien DNA, the vector needs to have the very few, preferably single recognition sites for the commonly used restriction enzyme. मतलब अगर एक से ज़्यादा साइट्स हैं तो बहुत मोर देन वन साइट्स पे वो कट लगाएगा और बहुत सारे फ्रेगमेंट्स जनरेट होंगे, एंड वी डोंट वांट दिस टू हैपन बिकॉज इट विल कॉम्प्लिकेट द प्रॉब्लम our genetic engineering process. So it needs to have preferably a single recognition sites for the commonly used restriction enzymes. For different restriction enzymes, it may have different sites or many other sites, but for the same or the commonly used restriction enzyme, it should have only one. Let's suppose there is a restriction enzyme called A, then A should have only one site. It may have sites for A, B, C, D, but for A only one site, B only one site, for C also it should have only one site. Presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning process. So this ligation of alien DNA is carried out at restriction site present in one of the two recombinant resistant genes. So selectable markers हैं जो antibiotic resistant gene हमने डाली है, that helps us to identify the recombinants from the non-recombinants, can only work when we introduce the gene in one of those antibiotic resistant genes. So अगर यहाँ पे डायग्राम में उन्होंने दिया है PBR322 नाम का एक वेक्टर प्लाज्मिड है. So it has two antibiotic resistant gene. One is ampicillin resistant gene, that is this one which I have marked with blue, and the other one is tetracycline resistant gene marked with this red. The one which is underlined with the blue is ampicillin and the other one is tetracycline. Okay, the ampicillin resistant gene has two sites for two different restriction enzymes. One is PST1 and other is PVU2. And the tetracycline resistant gene has again two sites for two different restriction enzymes. One is PAM H1 site and other is cell H1. Cell 1 site. Then there is ORI, that is origin of replication. It is a sequence from which the origin of replication begins. Behind it, there is a ROP, that is replication of plasmid. It is a site for restriction enzyme PBU2. So function of ROP is it helps in replication of plasmid and it codes some proteins that are required for the replication of plasmid. So in this para they have given that uh, replication of plasmid ROP codes for the proteins involved in the replication of the plasmid. That's why it is called as the ROP. The code for protein involved in replication of plasmid. Then besides this, there are three more sites between these two antibiotic resistant genes. One is for EcoR1, other is CLA1, and the third one is HIND3. The first restriction endonuclease discord was the HIND2, and this one is HIND3. Both are different. For example, if you ligate a foreign DNA at the PAM-H1 site of tetracycline resistant gene in the vector PBR322, the recombinant plasmid, that is the plasmid having that introduced gene, are called as the recombinant plasmid. They will lose tetracycline resistance due to the insertion of foreign DNA. जो आपने फॉरेन डीएनए उनमें डाला है उस जीन में टेट्रासाइक्लिन रेजिस्टेंट जीन के बीच में, so that's why it will lose the resistance due to this insertion of the foreign DNA, but it can still be selected out from non-recombinant ones by the plating the transformants, that is the bacteria having this plasmid on the tetracycline containing medium. Transformants are those which have this plasmid, but transformants may be recombinants or non-recombinants. Recombinants मतलब those transformants in which the foreign DNA is present, and non-recombinants are those transformants in which foreign DNA is absent. So जिसमें foreign DNA present है, it will show resistance only towards the ampicillin because it will lose the tetracycline resistant. But the other one in which the non-recombinant one will show resistance towards both of them because both of them are functional, and the transformants growing on the ampicillin containing medium are then transferred on a medium containing tetracycline. मतलब जो transformants growing on ampicillin containing medium है, on ampicillin containing medium they will survive because they have ampicillin resistance gene in them which is functional. But when you transfer them on tetracycline containing medium, the recombinants will grow in ampicillin containing medium but not on the tetracycline because recombinants lose the tetracycline resistance due to foreign DNA. But non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistant gene helps in selecting the transformants, whereas the other antibiotic resistant gene gets inactivated due to the insertion of alien DNA and helps in selection of the recombinants. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from the non-recombinants on the basis of their ability to produce the color in the presence of chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of enzyme beta-galactosidase. There is an enzyme called beta-galactosidase which codes for blue color. So when the DNA is inserted within the coding sequence of the enzyme, it becomes inactive and results into the inactivation of gene for synthesizing of this enzyme, which is referred to as insertional inactivation. The presence of chromogenic substrate gives blue-colored progenies or the colonies if the plasmid in the bacteria does not have the insert. If you don't insert the foreign DNA, it will still show the blue color. But presence of insert, so that is after inserting the foreign DNA, after insertional inactivation, it results into it will not show blue colored progenies anymore. This is called as insertional inactivation, and they are identified as the recombinant colonies because they have the insert in them. Vectors for cloning genes in plants and animals. You may be surprised to know that we have learned this lesson of transferring the genes into the plants and animals from bacteria and viruses which have known for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria or viruses want. For example, Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as the tDNA to transform normal plant cell into a tumor cell. So what is the DNA which the Agrobacterium tumefaciens, which is a dicot pathogen, has? It is called a tDNA. So mark this. Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as the tDNA to transform normal plant cell into a tumor and direct these tumor cells to produce the chemical required by the pathogen. Similarly, in animals, we use the retroviruses. So retroviruses are actually, in animals, they have the ability to transform the normal cells into a cancerous cell. And a better understanding of the art of delivering genes by pathogens is the eukaryotic host has generated the knowledge to transform these tools of pathogens into a useful vector by delivering genes of interest to humans. The tumor the tumor inducing plasmid of Agrobacterium tumefaciens, which has a tDNA, the tumor inducing means Ti plasmid, has now been modified into a cloning vector which is no more pathogenic. So it does not transform the normal cells into tumor anymore because it has been modified into a cloning vector, and it is still able to use the mechanism to deliver the genes of our interest into the variety of plants. Similarly, retroviruses have been disarmed and are now used to deliver desirable genes into an animal cell. In case of plants, we use Agrobacterium tumefaciens, which has the tumor inducing plasmid. In case of animals, we use retroviruses to deliver the desirable genes into the animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host. Now let us come to the competent host for transformation with the recombinant DNA. Since DNA is hydrophilic molecule, that is, hydrophobic means water, the one which repels water is hydrophobic and one which attracts water is hydrophilic. So DNA is a hydrophilic molecule. It attracts water, but it cannot pass through the cell membrane. Why? Because cell membrane, it has outer hydrophilic part, but inside it is hydrophobic. So hydrophilic molecules, they find difficult to pass through the hydrophobic part. In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up the cell. So हमें उनको पहले तैयार करना होगा DNA उनके अंदर डालने से पहले. This is done by treating them with a specific concentration of divalent cations such as calcium. So to make bacterial cells competent, we treat them with a specific concentration of divalent cations like calcium, which increases the efficiency with which the DNA enters the bacterium through the pores in its cell wall. जो बैक्टीरिया के सेल वॉल में पोर्स होते हैं, उनसे वो DNA अंदर जाता है सेल में. So recombinant DNA can then be forced into the cells. Now after this, treating with specific concentration of divalent cation and increasing the efficiency of the uptake of DNA through the transient pores present in its cell wall, now they are forced further into such cells by incubating the cells with the recombinant DNA on ice. So we have to then incubate these bacterial cells with which are having the recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius. This is called heat shock. Suddenly you have to change them from very low temperature to very high temperature and then again put them back on ice. So from low temperature to very high, then again very low. From first incubate on ice, then heat shock at 42 degrees Celsius, then again ice. This enables the bacteria to take up the recombinant DNA. This is for bacteria. This is not the only way to introduce the alien DNA into host cell. In case of plant cell, in case of animal cell, we use microinjection. For bacteria, we have to treat with the divalent cation and then incubation and then heat shock. But in case of animals, we use a method known as microinjection. in which a recombinant DNA is directly injected into the nucleus of the animal cell. जो recombinant DNA और the plasmid हमने बनाया है वो हमें directly nucleus of the animal cell जो होता है उसमें inject करना है. This is for animal cells. Another method suitable for the plant cell is chain gun or the bioelastics. In this method, what happens is uh, this method is suitable for plants, and cells are bombarded with a high velocity microparticles of gold or tungsten metal coated with DNA, and this method is known as bioelastics or gene gun. उसको गन बोलते हैं क्योंकि जो पार्टिकल्स होते हैं माइक्रो पार्टिकल्स ऑफ गोल्ड और टंगस्टन दे एक्ट्स लाइक अ बुलेट. So they are coated with the DNA, and then they are inserted into this. The cells are bombarded with these high velocity microparticles of gold or tungsten coated with DNA. This is known as bioelastic or gene gun. This is suitable for plants. For animals, micro injection. For plants, bioelastics or gene gun. And the last method is the one which we have already discussed in the previous section, that is use of disarmed pathogens. Disarmed pathogens are those vectors which, when allowed to infect cell, they transfer recombinant DNA into the cell. They don't affect them, they don't cause any diseases because they are disarmed, like Agrobacterium tumefaciens, which is a pathogen of dicot plant for plants, and for animals we use the retrovirus. They also do the same job of transferring the DNA into their host cells.